Hey everyone, and welcome to our video on liver function. What we're going to be doing is carrying on with our module 5 work, and obviously the section all about those excretions of various waste products, and we're having a look at specification reference 5.1.2b part 1, the functions of the mammalian liver. Now, first thing we need to recall about the liver in terms of its function is that it's got a lot of different metabolic functions that it carries out for the body. So first one that we've got is that control of blood glucose that we've dropped into back in GCSE even, but it also helps control amino acid and lipid levels. It's going to be involved in the breakdown of various items like hormones, red blood cells, and certain chemicals, etc. that get into our body in that detoxification role. And then it also helps with the storage of certain key substances like some vitamins, like vitamin A, and also that glycogen, which ties into that control of blood glucose. There's also a bit of a synthesis role that it will carry out in terms of bile, plasma proteins, and cholesterol as well. So when it comes to the liver itself, it's got this diverse range of different metabolic functions which are critical to the survival of that organism at the end of the day. Let's start with the storage role. Now, even back at GCSE, we were talking about the role of the liver as a storage organ for that blood glucose control. So what we find is the liver is actually going to store sugars in the form of glycogen, and this glycogen will be stored in granules. And those granules will be located within our hepatocytes. And remember from our previous video, hepato is basically meaning liver, and the word cyte means cell, so the liver cells. And it's the cytoplasm of those hepatocytes that we will find these granules containing that glycogen. This is obviously a good place that we can see a link back to our module 2 work on our biological molecules, because back in module 2, one thing that we did was we had a look at carbohydrates. So you had a look at the key kind of various monosaccharides, and then we had a look to see how we can form the polysaccharides, glycogen being one of them. So look out for those linking questions taking us back to our biomolecules when we are talking about the liver as well. The second role that we're going to focus on is the detoxification role. Now, what we find is that within our body, we will have certain substances that either we have ingested or that are created that can cause us harm if they're allowed to remain inside us. Therefore, we've got to have some kind of facility to remove these harmful substances. And the liver is the site of this detoxification of a range of them. And in terms of the way that it's going to actually remove these toxins, there's 4 kind of key methods you should be aware of: oxidation, reduction, methylation, and combining it with another molecule. So obviously we've got a range of different ways in which that we can basically make these toxins less problematic by making them harmless to us. And therefore, depending on which toxin we are talking about, would obviously select the relevant method. The good news is you don't need to know vast quantities about each of those procedures. Just know oxidation, reduction, methylation, and combining with another molecule are ways that we can render these toxins harmless to the organism. What we are going to do though is have a little look in a bit more detail at one particular example of detoxification in a moment. But before we can really delve into that example in more detail, we should be aware of why the liver is able to detoxify this range of chemicals that it may have inside the organism. And the key thing is that in those liver cells, the hepatocytes, we actually find lots of enzymes. And I've given you a nice example here, catalase. And catalase is an enzyme you may well have encountered in practical work in biology. It's all to do with increasing the rate of breakdown of hydrogen peroxide. Now, hydrogen peroxide is a harmful substance, and you may encounter it in your everyday life. If you like to dye your hair, hydrogen peroxide is going to be there. In an organism, it's not going to be good to have an accumulation of hydrogen peroxide, so we need to break it down. And it does break down naturally, very slowly, into just oxygen and water to harmless substances. but we don't want it to break down slowly. We need to increase the rate, hence catalase. So catalase actually has this turnover number of 5 million. Now, turnover number quite simply just tells us how many molecules of that substance can be broken down by one catalase molecule each second. So if we've got a turnover number of 5 million, it's pretty quick. Okay, so that means that when we've got that hydrogen peroxide, the catalase is able to convert it into the water and the oxygen really quickly. 5 million molecules each second per catalase. Second little example I'm going to give you is cytochrome P450. Now, cytochrome P450 isn't a single enzyme, this is a group of enzymes, and these are going to be involved in breaking down various drugs, and the reason that I'm talking about them here is because they're going to pop back again when we have a look at the electron transport chain. So what we're seeing here is this cytochrome P450, this group of little enzymes. They're not only involved in breaking down drugs, but also in the electron transport chain, which we're going to see in processes like respiration, for example. So our specific example we're going to focus on is alcohol. Now, alcohol is a depressant, and what we actually find is obviously it's something we consume, and therefore it's going to be absorbed into the bloodstream through the stomach. Really quite readily. It's very easily absorbed into our bloodstream. Now, because it's then going to get into our bloodstream, it's going to be distributed pretty quickly through the body, and when it gets to the liver, will be absorbed by the hepatocytes, the liver cells. Now, you might think, okay, we're going to take this alcohol in, let's just break it down and get rid of it because it's harmful to us. We don't want it accumulating. But it's also a really good source of energy. And as always with living things, we don't really want to waste potential sources of energy. So what we're going to actually see happening as far as the alcohol goes is that the ethanol is going to be respired by those hepatocytes. So how does this happen then? Well, first of all, we've got to undergo a couple of steps to convert the ethanol into something that can feed into respiration. So we start off with ethanol, C2H5OH, hopefully our GCSE chemistry lets us know that, and we're then going to convert the ethanol into ethanal, and that's all going to be conducted through the use of an enzyme, ethanol dehydrogenase. Now, if we have a look at this enzyme dehydrogenase, so we've got the word hydrogen in there, and we've got D at the front. So a dehydrogenase enzyme is basically going to be removing hydrogens from the substance the first word is, in this case ethanol. So basically we have ethanol, we're going to remove hydrogens from it, and therefore we form ethanal. And those hydrogens that we've removed from the ethanol are actually going to be acquired by our NAD, and when hydrogen joins with NAD, we form reduced NAD. So our first stage then is that we have got ethanol having two hydrogens removed through the process of that ethanol dehydrogenase to form ethanal and reduced NAD. Word of advice. Obviously we can start writing our reduced NAD in terms of NADH, etc., but you are going to find that we are going to be looking at NAD, NADP, we're going to be looking at FAD, and all of these are going to be reduced by the addition of hydrogen or hydrogens. If you're trying to write these on your A-level biology paper, you could very easily mix them up. Put the 2's in the wrong places, forget the 2, etc. Why run the risk of doing something silly when you will equally get the mark just for writing reduced NAD? Make life easier for yourself. Don't try and remember all the different formulas. Just literally shove the word reduced in front of whatever chemical we have reduced. The second step to this is we are then going to take our ethanal and we're going to dehydrogenate it using ethanal dehydrogenase this time. And this time we form ethanoate, or the old name used to be acetate. So depending on what resource you're looking at, could be either of those two. So we can see that in our little pathway at the bottom. We're starting off with the ethanal. We've got our ethanal dehydrogenase removing a couple of hydrogens to form the ethanoate, and those hydrogens again are going to form reduced NAD. Once we've actually got this acetate or the ethanoate, then that's going to combine with this substance coenzyme A. And that means that we're going to form acetyl coenzyme A. And when we come on to respiration, you're going to see this because it's going to reoccur there. That acetyl coenzyme A is then going to enter our aerobic respiration pathway at the point of the Krebs cycle. So basically, what we've been able to do here is take the ethanol, which is a harmful substance, and through a couple of steps, we've then changed it into the acetyl coenzyme A, which can enter Krebs cycle and therefore be respired to provide us with energy. So here's the whole pathway, just so that we've got it all as one easy reference little slide. Start with ethanol. Ethanol dehydrogenase removes a couple of hydrogens which are attached onto NAD to form reduced NAD. We also get the ethanal. Then ethanal dehydrogenase dehydrogenates the ethanal to form ethanoate and reduced NAD. And finally, the ethanoate is going to combine with the coenzyme A to form acetyl-CoA, which joins Krebs cycle. In terms of why this is important, not only have we got the acetyl-CoA going into Krebs cycle, but that reduced NAD that we formed at those first two stages are actually going to be used in the mitochondria to generate ATP as well. So what we're going to see here is we're actually able to generate certain quantities of ATP from the ethanol that we took into our body in the alcoholic drink. Now, as a result of this, because if we're consuming ethanol, then our body is going to be utilising it to provide the ATP that is required by those cells, the liver cells won't be using fats in order to generate their ATP. So they're not going to be metabolising those fats. As a result, we're going to have this excess of fat basically, and that gets stored in those hepatocytes, and that means we end up with a condition called fatty liver. So anytime that we've got this fat stored in hepatocytes, those hepatocytes then lose their efficiency, and over time, if you keep doing this, you can eventually develop that medical condition of cirrhosis. The other key role of the liver that we're going to look at in more detail here and again later on in our Module 5 work is looking at how we actually produce urea. Now, in terms of our basic diet, we need between 40 and 60 grams of protein a day, but quite frequently the amount of protein that we consume actually exceeds this. Now, because of that, obviously proteins, back to our Module 2 work again, are made up of amino acids. And therefore, as we digest those proteins into their constituent amino acids, we then get an increased number of amino acids in the body. And the problem that we find with this is that the amino groups on those amino acids are actually toxic, so we can't store those excess amino acids that we have. Again, we do not want to just basically get rid of it all because that would be a waste of potential energy, and therefore what we're going to do is basically release that energy in this two-stage process in the liver. So stage 1 is a process we call deamination. And basically what we're going to do here is we start with our amino acid, which we can see on the end here, and we're going to remove the amino group and therefore result in two products, which will be the keto acid and ammonia. Now, what we do need to bear in mind is that ammonia isn't actually much better than the amino acids themselves, therefore this is only the first stage. So stage 1, deamination. Amino acids are converted into a keto acid and ammonia through the removal of that amino group. Now, the ammonia itself, as we mentioned, is actually highly soluble and toxic, so we've got to get rid of that as well. The other aspect though, the keto acid, this is actually the useful bit. We can then put the keto acid through the process of respiration and therefore utilise the energy that's stored within the molecule. Alternatively, we could take that keto acid and convert it into a carbohydrate or fat, and then we could store that chemical. The second stage that's going to deal with the ammonia that we've now created is the bit called the ornithine cycle. So what we're going to see here is that ammonia is going to combine with carbon dioxide and ornithine, which is just an amino acid, and we produce this stuff called citrulline. Now the citrulline leaves the mitochondria through the process of facilitated diffusion, and then we're going to have ammonia added to the citrulline to make arginine. Arginine can then go back to ornithine by having urea removed, hence the ornithine cycle. We start with ornithine, and then we're also going to basically end with ornithine. So all we're doing is we're going through a little kind of multiple step conversion here to take the ammonia, join it with carbon dioxide and ornithine. We go to citrulline. Citrulline then is converted to arginine, arginine back to ornithine, and we're able to remove that urea. So this is basically what it looks like. So we've got our nice little ornithine here. We've got obviously the ammonia here, and we add some carbon dioxide to it. So those three components come together and then form the citrulline. What then happens is we can add more ammonia, and that means that we're going to convert the citrulline to arginine, and then arginine is converted back to ornithine with the removal of that urea. The key reason that we're going to do this is that urea is less soluble and less toxic than ammonia, and what's going to happen there is the urea is then able to pass into the blood, and when it reaches the kidneys it will be filtered out, and then it will end up in the bladder where it will be stored as urine until hopefully you reach a toilet. As always, I hope you found this useful, and obviously make sure you subscribe to the channel so you can see when I next upload a video, and head on over to the website where you can find a range of other resources to help you in your A-level biology studies.